after successfully completing the part 1, that is chapter 5 in the second PUC, so principles of inheritance and variations, let me start with the second part. So second part begins with the chromosomal inheritance. So chromosomal theory proposed by Sutton and Bowen. Prior to this, so John Gregor Mendel published his works in 1865. But till 1900, his works did not get any recognition at all. This is because of the 4 reasons. Because John Gregor Mendel's work did not published internationally. That is one reason. And John Gregor Mendel introduced mathematics to the field of biology, which was totally unacceptable in those days, unacceptable to the scientists of those days, the introduction of mathematics to the field of biology by John Gregor Mendel. That is the second point. And John Gregor Mendel described gene as the unique stable factors, which was not correct. Stable factors, he called these genes as stable factors, and he was not aware of the chemical composition of the genes. These are the 4 points. Because of this, it was unrecognized till 1900, though John Gregor Mendel published his works in 1865. So next to this, the chromosomal theory proposed by Sutton and Bowen. Proposed the chromosomal theory, and we know that the variations occur in organisms because of the cross fertilization, especially during the crossing over. The exchange of genetic materials takes place. That's also very important. Sutton, by combining the Mendelian laws and his theory, he proposed one more theory called chromosomal inheritance theory. That is by Sutton. And the T.H. Morgan, he did experiments on the Drosophila melanogaster. Drosophila melanogaster, commonly called fruit fly, Cinderella of genetics. So many geneticists, they selected only these Drosophila for their experiments because of 5 reasons. Number 1, the lifespan will be very less, so that we can study many generations. Lifespan will be very less, that is only 2 weeks to 1 month, and a single mating can produce a large progeny. And culturing medium, that is very simple culturing medium. We can observe the variations in these even through the low microscope, and sex can be determined easily. These are the 5 points. Because of this, so many experiments did on Drosophila melanogaster by the scientists, and hence Drosophila melanogaster called the Cinderella of genetics. So the T.H. Morgan, Thomas Hunt Morgan, noticed the linkage. That means the characters are inherited from parents, linked together, stayed together, linked together, stayed together, called linkage. The linkage. And the Alfred Stuyvesant used the frequency of recombination between gene pairs in the same chromosome as a measure of distance between genes, and mapped their positions on the chromosomes. So this kind of chromosomal mapping also plays an important role in Human Genome Project that we will study later on in the other next chapter, Human Genome. So this kind of chromosomal mapping based on the distance between the genes that was measured by the Alfred Sturtevant, student of T.H. Morgan. So if the genes are closely packed, recombinant types will be very less and the parental types will be very more. A cross between yellow-bodied white-eyed Drosophila with brown-bodied red-eyed Drosophila. What happens here? The genes are closely packed, hence the parental type is about 98.7%, but the recombinant very less, 1.3%. If the genes are distantly arranged far away from the other one, so the recombinant will be more, and so this we can observe in a cross between the Drosophila white miniature. That means here white means white-eyed, miniature means with small wings, miniature, small wings. This is very important, and that is with normal wings with red eyes. Here, as the genes are distantly arranged, the genetic recombination will be 37.2 and the remaining 62.8 of parental type. So, if the genes are closely packed, the linkage will be more, the parental characters are inherited, and the recombinant will be less. And if the genes are distantly arranged, recombinant characters will be more, and the linkage will be reduced. So this is about the linkage. Next, the sex chromosomes. So the X body, Henkin noticed the X body in insects. So the sex chromosomes are, in case of human beings, XX in females and XY in the males. So females are the homogamity and the males are heterogamity, because the males produce two types of sperms: a sperm with X chromosome, that is 22 autosomes, and a sex chromosome 22X, and sperms with 22Y. So thus the males are heterogamity, and the females homogamity. They produce only one type of gamete, that is with 22X in case of human beings. So sex is determined by, whether the male or female will be determined by the sex chromosomes. In case of insects, the females are homogametic, XX they have, but the males are heterogametic, X and 0, that is O type. One sperm with X chromosome and one sperm without any chromosome, O type. So here, if the sperm carrying X fuses with the egg, the resulting zygote develops into the female in insects, in majority of the insects. And if the sperm without any X chromosome, that is O type, and fuses with the egg cell, that is X, the resulting zygote develops into male, that is the sex determination in case of many insects. Whereas in case of Drosophila, the females are homogametic, XX, but the males are heterogametic, just like human beings, XY, where the 2n equals only 8 in Drosophila melanogaster. If the sperm carrying X fuses with the egg, the resulting zygote develops into female Drosophila. If the sperm carrying Y fuses with the egg, the resulting zygote develops into the male Drosophila. Similarly, in case of human beings also, the females are homogamity, they exhibit homogamity, and the males heterogamity. If the sperm carrying X fuses with the egg, the resulting zygote develops into a girl. If the sperm carrying Y fuses with the egg, the resulting zygote develops into a boy. And a peculiar type of this sex determination can be noticed in case of the birds. The birds is an exception, and here the males exhibit homogamity, that is ZZ sex chromosomes males have, and females have ZW heterogamity. And here the egg determines the sex, the type of egg. That is, the egg if carrying the Z type of chromosome fused by the sperm of Z of homogamity, then the resulting zygote ZZ develops into male. And if the egg having W fused by the sperm of ZW, that zygote develops into female. Thus, the heterogamity will be exhibited in birds. That is very important for your NEET. And sex determination in honeybees. This is very, very important. 2n equals 32. A bee colony consists of about 60,000 to 80,000 honeybees. Only one queen, which is fertile, that is 2n equals 32. It has chromosomal number. And there are about 200 to 300 males called drones. They are haploid. Their chromosomal number equals 16. And the females, the 2n equals 32. They are sterile. They are sterile. But males are fertile, even though they have the 16 chromosomes. And the female queen is fertile, 2n equals 32, and it produces eggs, and it also receives sperms from the male drones, and sperms are stored in spermatheca. Sometimes eggs are laid, simultaneously sperms are released. The fertilized eggs develop into the females, but they are sterile, that is the female worker bees. Sometimes eggs are laid and sperms are not released. These eggs directly develop into the males, which are haploid. There, the chromosomal number is 16, and the development of offsprings without fertilization is called parthenogenesis. This kind of sex determination takes place in case of honeybees. It is a matter of fertilization and no fertilization. That is the thing that determines sex in case of the honeybees, and this is known as parthenogenesis. In plants, if the ovary develops into fruit, that is called parthenocarpy. This is very important. Parthenocarpy in plants, already asked in the NEET many times. And parthenogenesis, that is in animals. 4 examples given in your book: RTHL, the code word, rotifers, turkey birds, honeybees, lizards. They exhibit the parthenogenesis. After this, mutation. Mutation is sudden change in the hereditary material which is heritable. That is, hereditary materials are chromosomes called vehicles of heredity. Chromosomes made up of DNA and histones. The DNA is the chief hereditary material. DNA made up of genes. Genes are called units of heredity. Hence, the chromosomes, DNA and genes are the hereditary materials. So, 2 types of mutations can be seen: gene mutation and chromosomal mutation. Gene mutation, natural gene mutation observed by T.H. Morgan, that is the appearance of the red eyes in Drosophila melanogaster, which is an example for natural mutation. And artificially, mutation can be induced by using agents called mutagens, which is very important. X-rays, UV rays are the things, mutagens, which bring about the mutation artificially. So, gene mutations, 2 types: natural gene mutation and artificial gene mutation induced by the mutagens. And chromosomal mutations, also 2 types: numerical mutations and the structural aberrations. So, any structural aberrations or numerical aberrations bring about cancer, especially the numerical aberrations of the chromosomes bring about cancer. Idu kelila, idu important. So, structural aberrations ಅಥವಾ mutations in aberrations ಅಂತ ಕರೀತೀವಿ. Structural aberrations in chromosomes due to deletion, duplication, inversion, translocation. Deletion, a part cut ಆಗುತ್ತೆ. Deletion, duplication, repeat ಆಗುತ್ತೆ. Inversion, ತಲೆಕೆಳಕಾಗಿ ಕಟ್ ಆಗುವಂಥದ್ದು inversion, translocation. This kind of change in structure, chromosomal structural aberrations in chromosome, because of deletion, duplication, inversion, translocation, bring about mutation. ಇದು ಕೇಳಿಲ್ಲ, ಇದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. And the numerical mutations, 2 types: aneuploidy and euploidy. Aneuploidy means ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಇವೆಲ್ಲ. Aneuploidy means change in number because in the chromosomes, because of addition or loss of one or two chromosomes. Addition or loss ಆದರೆ ಅದನ್ನ aneuploidy ಅಂತ ಕರಿತೀವಿ. Aneuploidy. So loss of one chromosome called monosomy. Example, Turner syndrome ಹೇಳ್ಬೋದು. And addition of an extra chromosome, maybe autosome or sex chromosome, that is called trisomy ಅಂತ ಕರಿತೀವಿ. ಟ್ರೈಸೋಮಿಗೆ ಎಕ್ಸಾಂಪಲ್ ಡೌನ್ ಸಿಂಡ್ರೋಮ್ ಅಂಡ್ ಕ್ಲೈನ್ಫೆಲ್ಟರ್ ಸಿಂಡ್ರೋಮ್ ನ ಮೆನ್ಷನ್ ಮಾಡಬಹುದು. ಡೌನ್ ಸಿಂಡ್ರೋಮ್ ಈಸ್ ಟ್ರೈಸೋಮಿ 21 ಅಂತ ಕರಿತೀವಿ. ಸೊ ದಿಸ್ ಈಸ್ ಅಬೌಟ್ ದ aneuploidy. Aneuploidy is change in number, that is the numerical aberration in chromosomes, that is because of addition or loss of one or two chromosomes. So this is about aneuploidy. And coming to the euploidy, it is a numerical aberration in chromosome. This is because of addition or loss of set of chromosomes, n number of chromosomes. For example, all the organisms consists of 2n, that is diploid condition, 2n represented by 2n. But some organisms contain more than 2 sets of chromosomes like triploid, tetraploid and so on. The phenomenon called polyploidy. Polyploidy first observed by Hugo de Vries in a plant called Eunothera lamarckiana, an evening primrose. ಇನ್ನೊಂದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಏನು ಅಂತ ಹೇಳಿದ್ರೆ, polyploidy is caused because of failure of cytokinesis. ಸೈಟೋಪ್ಲಾಸಮ್ ಯಾವಾಗ ಡಿವೈಡ್ ಆಗಲ್ಲ, ಅವು ಕ್ರೋಮೋಸೋಮ್ ಸ್ಟೇಟ್ ಟುಗೆದರ್ ಹೊರಟೋಗುತ್ತೆ. ಇನ್ಕ್ರೀಸ್ ಇನ್ ನಂಬರ್ ಆಫ್ ಸೆಟ್ ಆಫ್ ಕ್ರೋಮೋಸೋಮ್ಸ್ ಆಗುತ್ತೆ. ಅಂಡ್ ಪಾಲಿಪ್ಲಾಯ್ಡಿ ಇಸ್ ಬಿಕಾಸ್ ಆಫ್ ಫೈಲ್ಯೂರ್ ಆಫ್ ಸೈಟೋಕೈನೆಸಿಸ್ ಅನ್ನೋದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಆಗಿರುತ್ತೆ. ಅಂಡ್ ದೆನ್ ಇದರಲ್ಲಿ ಏನಾಗುತ್ತೆ, ಡಿಲಿಷನ್ ಅಂಡ್ ಇನ್ಸರ್ಷನ್ ಆಫ್ pairs of DNA causes frame shift mutation. ಅಥವಾ addition, insertion, deletion of pairs of genes of DNA causes frame shift mutation ಅಂತ ಕರಿತೀವಿ. ಅದನ್ನ ಚೆನ್ನಾಗಿ ಜ್ಞಾಪಕ ಇಟ್ಕೋಬೇಕು. ನೆಕ್ಸ್ಟ್ ಇಲ್ಲಿ ಕೊಡುವಂಥದ್ದು ಆಫ್ಟರ್ ದೀಸ್ ಡಿಫರೆಂಟ್ ಟೈಪ್ಸ್ ಆಫ್ ಮ್ಯೂಟೇಷನ್ಸ್, ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್ ಅಂತ ಕೊಡ್ಬೇಕು. ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್. And then the inheritance of characters or traits from generation to generation, tata, muttata, hige, from generation to generation, will be represented in the form of chart called pedigree analysis. ಅದಕ್ಕೆ ಎಲ್ಲ ಸಿಂಪ್ಟಮ್ಸ್ ಕೊಡ್ತಾರೆ. ನಾರ್ಮಲ್ ಆಗಿರುವಂಥದ್ದು, ಸೆಕ್ಸ್ ನಾಟ್ ನೋನ್ ಅನ್ನುವಂಥದ್ದು, ಅಫೆಕ್ಟೆಡ್ ನಾಟ್ ಅಫೆಕ್ಟೆಡ್, ಅದನ್ನ ಚಾರ್ಟ್ ಅದು ತೋರಿಸಿ ಅದರ ಮೂಲಕ ಅದನ್ನು ನೋಡಿ ಅದನ್ನ ತಿಳ್ಕೋಬೇಕು. ಹಿಯರ್ ಇದು ಬಹಳ ಒಂದು ಸಿಂಪಲ್ ಒಂದು ಇದನ್ನ ಹೇಳ್ಕೊಡ್ತೀನಿ. ಪೇರೆಂಟ್ಸ್ ಎಲ್ಲಾನು ನಾಟ್ ಅಫೆಕ್ಟೆಡ್ ಇದ್ರೆ ಮತ್ತೆ ಆ ಕ್ಯಾರೆಕ್ಟರ್ ಏನಾದರೂ ಇನ್ಹೆರಿಟ್ ಆಗಿದ್ರೆ, ರೆಸೆಸಿವ್ ಇರುವಂಥ ಪೇರೆಂಟ್ಸ್ ಆದರೆ ಕ್ಯಾರೆಕ್ಟರ್ ಇನ್ಹೆರಿಟ್ ಆಗಿದ್ರೆ ಅದು ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್. ಬಹಳ ತುಂಬ ಸುಲಭ. ಅವನು ಏನೇನೋ ವಿಪರೀತ ಕೊಡ್ತಾನೆ, ಆದರೆ ಪೇರೆಂಟ್ಸ್ನ ನೋಡ್ಬೇಕು. ಇಲ್ಲಿ ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್ ಎಕ್ಸಾಂಪಲ್. ಪೇರೆಂಟ್ಸ್ ಎಲ್ಲ ನಾಟ್ ಅಫೆಕ್ಟೆಡ್ ಇದ್ರೆ, ಅಲ್ಲಿ ಅಫೆಕ್ಟೆಡ್ ಇಂಡಿವಿಜುವಲ್ ಬಂದ್ರೆ ಅದು ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್. ಸೊ ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್ ಇವು ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ಅಲ್ಲಿ ಥ್ಯಾಲಸೆಮಿಯಾ ಇಸ್ ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್, ಸಿಸ್ಟಿಕ್ ಫೈಬ್ರೋಸಿಸ್ ಅನ್ನೋದು, ಸಿಕಲ್ ಸೆಲ್ ಅನೆಮಿಯಾ, ಫೀನಲ್ ಕಿಟೋನ್ಯೂರಿಯಾ, ಇವೆಲ್ಲ ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್ಗೆ ಎಕ್ಸಾಂಪಲ್. ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಅಲ್ಲಿ ಬರೀ ಸಿಕಲ್ ಸೆಲ್ ಅನೆಮಿಯಾ ಕೊಟ್ಟಿದ್ದಾರೆ, ಬಟ್ ನಾವಿನ್ನು ತಿಳ್ಕೊಳ್ಳೋಣ. ಥ್ಯಾಲಸೆಮಿಯಾ, ಸಿಸ್ಟಿಕ್ ಫೈಬ್ರೋಸಸ್, ಸಿಕಲ್ ಸೆಲ್ ಅನೆಮಿಯಾ ಕೊಟ್ಟಿದ್ದಾರೆ, ಫೀನಲ್ ಕಿಟೋನ್ಯೂರಿಯಾ, ಇವೆಲ್ಲನೂ ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್ ಆಗುತ್ತೆ. ಪೆಡಿಗ್ರಿ ಅಂದ್ರೆ ಅಲ್ಲಿ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಇರಲ್ಲ, ಆದರೆ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಬರುತ್ತೆ, ಅದು ಆಟೋಸೋಮಲ್ ರೆಸೆಸಿವ್. ಸೊ ಮೇಲೆ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಇದೆ, ಕೆಳಗಡೆ ಬಂದಿದೆ, ಅದು ಆಟೋಸೋಮಲ್ ಡಾಮಿನೆಂಟ್. ಒಂದೇ ಒಂದು ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತಾನೆ, ಮಯೋಟ್ರೋನಿಕ್ ಡಿಸ್ಟ್ರೋಫಿ. ಸೊ ದಿಸ್ ಇಸ್ ಅಬೌಟ್ ದ ಪೆಡಿಗ್ರಿ ಅನಾಲಿಸಿಸ್. ನೆಕ್ಸ್ಟ್, ಇನ್ ದ ಲಾಸ್ಟ್ ಆಸ್ಪೆಕ್ಟ್ ಆಫ್ ದಿಸ್ ಚಾಪ್ಟರ್, genetic disorders and chromosomal disorders. So the genetic disorders are thalassemia, color blindness, hemophilia, cystic fibrosis, sickle cell anemia, phenylketonuria. ಇವೆಲ್ಲನೂ ಏನ್ ಹೇಳ್ತಾನೆ ಅವನು, ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್ ಅಂತ. ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ಸ್, ಡೌನ್ ಸಿಂಡ್ರೋಮ್, ಟರ್ನರ್ ಸಿಂಡ್ರೋಮ್, ಕ್ಲೈನ್ಫೆಲ್ಟರ್ ಸಿಂಡ್ರೋಮ್, ಅವು ಕ್ರೋಮೋಸೋಮಲ್ ಡಿಸಾರ್ಡರ್ಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಸೊ ಡಿಸಾರ್ಡರ್ಸ್, as I told you, it is because of mutation ಇರ್ಬೋದು. So that is a deletion or mutation ಇರುವಂಥದ್ದು. ಸೊ ಫಸ್ಟ್ ಒನ್ ನಿಮಗೆ ಅಲ್ಲಿ ಕೊಟ್ಟಿರುವಂಥ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್ ಯಾವುದು ಅಂದರೆ ಫೀನೈಲ್ ಕೀಟೋನ್ಯೂರಿಯಾ. ಫೀನೈಲ್ ಕೀಟೋನ್ಯೂರಿಯಾ ಜೆನೆಟಿಕ್ ಡಿಸಾರ್ಡರ್. ಇಟ್ ಈಸ್ ಇನ್ಹೆರಿಟೆಡ್ ಥ್ರೂ autosome recessive genes, autosome recessive traits. So you ಎರಡು ಪಾಯಿಂಟ್. In case of phenylketonuria, phenylalanine is not converted into tyrosine. ಸಾಮಾನ್ಯವಾಗಿ ನಮ್ಮಲ್ಲೆಲ್ಲನೂ ಟೈರೋಸಿನ್ ಕನ್ವರ್ಟ್. But in this case, phenylalanine not converted into tyrosine, but converted into phenyl pyruvic acid. Pyruvic acid ನಮ್ಗೆ ಗೊತ್ತಿದೆ CH3COOH ಅಂದ್ರೆ CO ಗ್ರೂಪ್. ಕೀಟೋನ್ ಇರುತ್ತೆ. So, because of this results in mental retardation, and because of poor absorption by the kidneys, the ketone bodies appear in urine, hence called phenylketonuria. Ketone bodies, that is CO, phenyl pyruvic acid group. So these are the symptoms of this one, phenylketonuria, and it is an example of the one gene showing many phenotypes, that is pleiotropy. Pleiotropy idu kuda example agutte, that is phenylketonuria, PKU, where once again phenylalanine is not converted into tyrosine but converted into phenyl pyruvic acid, and as a result this increases the mental retardation and poor absorption. Because of poor absorption by the kidneys, these things, ketone bodies are excreted through the urine, which is a genetic disorder inherited through the genes of the chromosomes. So this is about phenylketonuria. Cystic fibrosa explanation kottilla, and other two examples ಏನು ಹೇಳ್ತಾನೆ, sickle cell anemia. Sickle cell anemia is a genetic disorder. First point, it is inherited through recessive genes of autosomes. ಇಂಥವೆಲ್ಲ ಕೇಳ್ತಾನೆ, recessive genes ಯಾವುದು, autosomes ಯಾವುದು ಅಂತ ಕೇಳ್ತಾನೆ. ಅದಕ್ಕೋಸ್ಕರ ತುಂಬ ಇಂಪಾರ್ಟೆಂಟ್. It is inherited through recessive genes of autosomes. That is very important. Both this one and even cystic fibrosa ಅದು ಕೊಟ್ಟಿಲ್ಲ. And then the thalassemia inherited through recessive genes of autosomes ಅನ್ನೋದು ಇಂಪಾರ್ಟೆಂಟ್. ಇಲ್ಲಿ ಏನಾಗುತ್ತೆ, sickle cell anemia, as the name itself indicates, they have abnormal RBC, sickle-shaped RBC. This kind of sickle-shaped RBC, it is because of abnormal hemoglobin. And the hemoglobin composed of 574 amino acids, adರಲ್ಲಿ 2 alpha chains, 2 beta chains. In the beta chain 6th position, if the hemoglobin contains valine in the place of glutamine, this is confusion, it's ತುಂಬಾ important. If it contains valine in the beta 6th position of chromosome, in the place of the glutamine, it contains valine, this is abnormal hemoglobin, HbS ಅಂತ ಕರೀತಾರೆ. So this change of the amino acid on the hemoglobin, it is because of change in codon on DNA. It contains GUG in the place of GAG. ಇದೆ GAG ಇರಬೇಕಾಗಿತ್ತು. GAG codes for glutamine and GUG codes for valine. So it contains GUG in the place of GAG. This change is because of change in nitrogen base on DNA. This contains CAC in the place of CTC. Because of this, the CTC and the CAC, ಒಂದು ಪಾಯಿಂಟ್ ಹತ್ರ mutation. This is known as point mutation, which is very, very important. And sickle cell anemia is of two types. The homozygous sickle cell anemia, 10% it causes anemia, and then the heterozygous, that is HBS and HBA, that is heterozygous sickle cell anemia. It causes only 50% anemia, and only HBS homozygous, that causes 10% anemia. So this is about sickle cell anemia, tumba important. Just like sickle cell anemia, another genetic disorder is thalassemia. Thalassemia is a genetic disorder which is invented through recessive genes of autosomes undoubtedly. So here the quality change, sickle cell anemia, it contains GUG in the place of GAG, it contains value in the, it's a quality change, but here quantitative change, quantitative mutation. The mutation results in the quantitative change, and number of globin chains which form hemoglobin will be reduced, especially HbA1 and HbA2 on the chromosome 16th. On the chromosome 16th, deletion of one or more of 4 genes takes place on the 16th chromosome, resulting in thalassemia. Thalassemia causes anemia, which is the quantitative change, unlike that of the sickle cell anemia. So this is about the thalassemia. Next to this, the 2 other genetic disorders are